Yeah, good evening, everyone. Welcome to today's CellTalk webinar. Today, our speaker, Dr. Vijay Padul, will be delivering a topic on all a comprehensive database for human aging and longevity studies. So, Vijay Padul is a senior scientist at RENEX who leads bioinformatics, genomics, and next generation sequencing research related activities in the organization. He is currently working on the neoantigen cancer vaccine pipeline projects. And Vijay holds a PhD in the life science from Omi Baba National Institute, Navi Mumbai. His PhD study focused on cancer genomics. Previously, Vijay has worked as assistant professor at graduate and postgraduate degree colleges as well. At RENEX, he has gained experience in scientific, operational, and administrative matters. I welcome Dr. Vijay to start the presentation.
Thank you, Chaya, for the introduction. For our today's Journal Club CellTalk activity, I have selected a paper that describes a database for aging and longevity related studies. So the paper title is HALL, a comprehensive database for human aging and longevity studies. The original authors are Hao Li et al. The title for the presentation is a comprehensive database for aging research. The topic selected is emerging technological advances in longevity and health span. The paper was published in journal Nucleic Acids Research with impact factor 16.6, and this paper was published in 2024. This research has an impact on aging research and longevity research. So coming to the introduction. So human aging and longevity studies, these are rapidly evolving. A lot of different aspects are being taken into consideration to study how humans age and what are the factors that are affecting the longevity. And there are different research groups that are carrying out research taking into consideration these different aspects, and therefore a huge amount of data is being generated by these studies in the recent years. And one of the major contributors to the increasing amount of data is the omics technologies. And because of their parallel nature and ability to provide multidimensional data, they have transformed the study of human aging, leading to the greater understanding of human aging. These data include Phenomics, Genomics, Transcriptomics, Proteomics, Metabolomics, Microbiomics, etc. All these technologies, they provide multidimensional insights, and also they have revolutionized the cohort-based research in understanding human aging and longevity. And also these data are very large in size. The accumulation of this data, this is coming from the study of different cells, tissues, cohorts across the lifespan of the individuals which were participating in those particular studies. And this necessitates a particular repository where all these data coming from different studies can be deposited and which can be accessed by the researchers world over. And because these different data, this is being generated from different life stages, such as from childhood to adulthood to the ultimate mortality of the individual. And these different stages, these are coming along because the body is undergoing different changes. And these are substantial transformations that are occurring in the human body. And therefore, these are large longitudinal studies which occur for longer amount of time. And therefore, the amount of data that is getting generated in single cohort studies are also very large. Particularly centenarians, the people that achieve 100 years of age, they have emerged as the potential models for the study of healthy aging. As because they have reached that particular certain age that exemplifies that they have all things essential that can ensure the healthy longevity in those individuals. And therefore, such studies are also important to record and to access for the future scientists. So individuals, they age at different rates and exhibit varying susceptibility to tissue aging and functional decline and aging-related diseases. And this is also a point of study to understand how different individuals age at different rates, because we can see that some people, they have higher biological age, but still they look younger, and also their functional age is also very younger. So therefore, because of this background, there is a pressing need for a comprehensive framework that can serve as a benchmark for monitoring human age-related changes. So this is a conceptual idea of this database, Human Aging and Longevity Landscape Database. So in this database, there are knowledge bases and multi-omics databases. The knowledge bases, they include genes and biomarkers related information about cross-tissue cross-tissue data, which can be specific and which can be applicable to the study of these biomarkers. The gene-related data, which are the genes which are affecting the aging and having impact on the longevity and also aging-related diseases. And these data, these are coming from different aging cohorts. And in this particular database, these cohorts are coming from 23 different countries and regions. And also there are more than 4.8 million samples have been added. And the age group of these individuals, it ranges from 1 to 119 years old. And there are more than 38 tissue and cell types involved. The multi-omics databases, they provide databases which include Phenomics database, Genomics database, epigenomics database, Transcriptomics database, Proteomics database, Metabolomics database, Metagenomics database, and Pharmacogenomics database. And all of these can be correlated or seem to have impact on the biological age, and this can be useful for cross-study analysis and also for the preparation of meaningful aging clocks. The present challenges for the establishment of or the availability of such databases is that till now there was a lack of established multimodal database, which focused on aggregating omics data coming from different cohorts and human tissues and age ranges, and with a specific emphasis on data related to human aging and longevity. There are certain existing databases, but they are not tailored for need to investigate human aging and longevity biomarkers and sensitivity to aging-related diseases. So therefore, there is a need for a comprehensive database. The objective of this particular research or activity was to establish a HAL database, which is a short form from Human Aging and Longevity Landscape Database, which is a comprehensive and multimodal web resource for the exploration of human physiology and pathology in all stages of the lifespan. So for creation of this database, the creators or authors, they manually collected data from literature, and also they explored different existing databases, and they collected all those things and created a comprehensive and reliable source of multidimensional database. This data incorporates 170 cohorts from 23 countries. There are more than 4.8 million individual data which is available here. The data is coming from 38 different tissue and cell types. The age range of individuals is 1 to 119 years of age, and there are 59 cohorts which contains centenarians as participants. So this is the structure of the database. So at the most higher level, this database has three parts: HAL for researchers, HAL for participants, and HAL for public. Each of these has different utilities for different user groups. So this is a website map of this HAL database. And after the welcome page, there are these three stages for level N: HAL for researchers, HAL for participants, and tools for the general public that they can use. Then HAL for researchers at level 2, level 3, and level 4, they provide different data. So first is cohort profiling, which enables searching for different cohorts that have already been carried out. So these cohort studies can be probed for a particular type of information if a researcher requires that. Then in the knowledge base, there are different level 3 data available, which provides data about genes and biomarkers. These databases being human aging knowledge, then human longevity knowledge, aging-related disease knowledge, human aging interventions, and from different publications in this area. Then there are multimodal databases available, which incorporate Phenomics, Genomics, epigenomics, Transcriptomics, Proteomics, Metabolomics, Metagenomics, Pharmacogenomics, etc. And for tools for researchers, they have provided a biological clock in which the existing biological clocks have been incorporated, and how this different data can be incorporated to create a biological clock. That information has been provided. Then in Hall for participants, participants meaning the research participants, if somebody wants to participate in a cohort study, which is studying aging and longevity, then people can participate by contributing their data. And here they can contribute their own data such as clinical parameters, and they can also see their how their performance is in terms of biological clock based on whatever clinical data and health information they have included into this particular database. Then they have also provided a biological L calculator tool where visitors can enter their essential health parameters, and this tool can calculate their biological age. This is a overview of Hall for researchers. So this Hall for researchers, it includes two uh, parts: knowledge bases and multi-omics databases. So knowledge bases, they include aging knowledge, and all these are providing data in terms of genes and biomarkers that are affecting the aging. Then longevity knowledge, aging related disease knowledge, aging interventions, and different publication in this area. Multi-omics databases, it provides data from different cohorts that is generating from this multimodal analysis: Phenomics, Genomics, Epigenomics, Transcriptomics, Proteomics, Metabolomics, Metagenomics, Pharmacogenomics. And these different aging cohorts, they have data range with age ranging from 1 to 117 years of age. So modules for researchers, it's cohort profile. It provides overview of major population studies that have already been collected and that are incorporated into the HAL database. Then there are multimodal databases which provide omics databases for aging and longevity studies. Then there is Phenomics module, and this is a curated list of phenotypic features which have been found to be closely associated with human aging and longevity. Then there is the Genomics module, which serves as a rich resource to view single nucleotide polymorphism organisms, that is SNPs, that are known to be associated with aging and longevity traits. Then there are epigenomics modules, which provides a powerful tool for querying DNA methylation alterations throughout the human lifespan. All the data that is available in the database can be probed through this module. Then there is the transcriptomics module, which provides integrative RNA-seq data sets of humans in all stages of their lifespan. Then single cell transcriptomics module is there, which serves as a rich resource for single cell molecular framework of human tissue and organ aging and human aging related diseases. Then proteomics module is there, which presents the proteome-wide atlases of age-associated alterations in humans. Then metabolomics module, it provides metabolome-wide atlases of age-associated alterations in humans. And there is a metagenomic module, which provides which reveals age and longevity-related alterations in human microbiota. Then the next part, HAL for participants, it enables volunteers participating in the registered clinical trials. The participants can provide information about their age-related characteristics. They can also provide assessments of their phenotypic age and biological age. Then the characteristics are calculated based on the diverse omic data that the participants will be submitting to the database. And there is also a feedback mechanism available that facilitates communication between the researchers and the volunteers. All these participants that will be participating in a particular study, if they have any questions or doubts, or they want to ask or give some information to the researchers who are conducting that study, they can directly contact through this particular module. Then there is HAL for public, so it provides access to biological age calculators to the public. So here phenotypic age and hormone age clock is available, which implements a phenotypic age and hormone age clock, and it requires to fill the user's form in a single format. And once they provide the required information, it provides insight into how their body functions and what is the correlation of their biological age with their chronological age. Then they also provide DNA methylation clock. This enables users to calculate DNA methylation age by uploading their DNA methylation data. And right now, only whole blood data is available option in the HAL database. So coming to discussion, HAL is a pioneering database dedicated to gathering data sets from diverse cohorts focused on aging and longevity. The HAL database vision is to leverage the aggregated data from HAL to expand the range of tools for assessing biological age. which encompasses various bodily systems such as nervous system and blood circulatory system, among others. So they're going to keep providing newer tools and newer analysis resources as they will expand this particular database. All it archives both types of studies cohorts, such as large scale cohorts and small scale cohorts, which encompasses a wide range of populations, including both genders and diverse ethnicities. So data from all over the world that were conducted in different studies, these have been incorporated into this database. HAL database, it also provides users with the ability to perform cross-cohort analysis. If somebody wants to compare different cohorts or study one single cohort in detail, it is possible to do that. And this will lead to the possibility to identification of common aging related features and also differences across different populations and regions, whether different populations have different kinds of aging pattern. These kinds of study can be performed by using these different databases comparatively. So this database also has limitations because of the intricacies of the individual health states. This tool right now serves only as an advisory tool to assess a person's biological age. That means whatever data they are using and whatever results they are showing to the public based on their different clocks, etc. They are cautioning that these are just advisory, and there might be some issues with the accuracy of the results that they have provided. In conclusion, this HAL platforms, it integrates the multimodal aging studies. It integrates participants, researchers, and tools, and creates an interconnected system which supports aging and longevity research for various stakeholders that will be interested in the aging and longevity studies. This linkage of these participants, researchers, and tools, it enables data integration across sources coming from different cohorts from different countries and different geographical regions. And this will assist in the biomarker research and intervention target identification with respect to aging and longevity. The impact of the research is that this platform empowers exploration of aging and longevity related changes and the identification of the association between multi-layered traits and aging dynamics. HAL's integration of participants, researchers, and tools creates an interconnected system for supporting aging and longevity research for various stakeholders. And indeed, this database, it fills a glaring gap between different cohorts that have already been conducted, and inability of researchers to getting the data in a single place. So this HAL database will be a valuable resource for researchers that are interested in study of aging and longevity. Thank you.
And thank you, Dr. Vijay, for your detailed talk. Now, floor is open for audience questions. Okay, I don't see any questions from the audience. Please move to the last slide. Before I conclude this webinar, I would like to inform all the next presentation will be given by Soumya Datta Basak on gene therapy mediated partial reprogramming, extends lifespan and reverses age-related changes in aged mice. Thank you for your kind attention and for joining today's webinar. We appreciate your time and engagement. We hope to see you in the next webinar. Thank you.